E1 or Lab 1. This is our very first lab. The way I'm doing this is I'll do lab recordings going over what we would do in a lab that we meet. These are the things that will be on quizzes and lab examinations. Then you'll do your Labster labs and all the video watching and things like that. So if you watch the course introduction video that I posted, then you kind of know what to expect there. So let's go ahead and get started on E1. This lab deals with basically microbes are everywhere. Kind of a little bit of pre-lab stuff where we talk about some of the basics of microbiology. Then I'll talk about microbes being ubiquitous or being everywhere. So let's get started. So microbiology, this is a big field of biology that deals with things you cannot see with your naked eye. You need to use a microscope, so we can't see with the unaided eye. So lots of people started off, we talked about in chapter 1 the history of microbiology, so you know loads of people besides microbiologists really started this, biologists, chemists, physicians, all kinds of people, but it's led to a lot of improvement to our longevity and quality of life within humans. So these things include, for microbes, this includes bacteria, which is a big center for this course. Archaea, that's the old things. They are prokaryotes, but not disease-causing. There are protists that cause diseases. There are fungi. And there's even some small things that are multicellular animals that cause diseases, all the way to worms and things like that. And then things we talk about which act as vectors. And then there's viruses that just aren't talked about here yet. Viruses, we never get to see a virus in here. We don't use electron microscopes, but we will get to see the impact of viruses on tobacco plants and things like that when we look at the slide tobacco mosaic virus. Now, most of actually things don't hurt us at all. Many are actually quite beneficial, even to the point it's essential that, for example, microbes make clotting factors, make vitamin K for clotting or things like that. But there are some that are disease-causing, pathogenic. And that's important, that they cause disease in humans and can be really harmful as well. So some there's everything from they don't hurt us to they really help us, we need them for our life, to some are harmful to us, and it's important that we know that. There is actually a lot of them we probably have not discovered. We probably haven't discovered all the microbes. We are trying to find them, going out there, hunting down microbes in, for example, the mountains of Appalachia to find some novel antibiotics, some new antibiotics for us, things like that. So what we'll do is we have prokaryotes, we have eukaryotes. Remember that this will always be on tests and quizzes. Prokaryotes, the word means pre-kernel or pre-nucleus. Pre-karyon basically refers to kernel, the nucleus. They are they lack membrane-bound nucleus. So that's a big thing, is they do not have a nucleus. They tend to be smaller than eukaryotes. They are simple. They lack membrane-bound organelles, so they're not going to have things like a Golgi apparatus, an endoplasmic reticulum, and all that good stuff. Their ribosomes are called 70S ribosomes, stands for 70 Svedberg units. The 70 Svedberg units, these are units of sedimentation. So that's what that is. That's what that stands for, Svedberg units. And they have circular chromosomes. Usually a bacteria will have a single circular chromosome. Some species of bacteria do have more than one chromosome, but most bacterial species have a single circular chromosome. Whereas in eukaryotes, they have multiple chromosomes that are linear. Both bacteria, archaea, that is our prokaryotes. Bacteria is going to be mostly what we focus on in microbiology, is bacteria, because that's what is easy to culture in laboratory settings. We will talk about some other things though. Eukaryote, eukaryon, eu meaning true, karyon basically meaning nucleus, refers to a kernel, referring to the true membrane-bound nucleus that they have. They're going to have the really interesting shapes. You guys remember learning about neurons, muscle cells, like skeletal muscle, cardiac muscle, smooth muscle, and how different the cells were, macrophages in A&P, and all these other different cells that we learned about in things like anatomy and physiology. There's all kinds of differences in size, shape, morphology, complexity of structure. There's a tremendous diversity in cells. They're going to have membrane-bound organelles, things like your Golgi, things like your endoplasmic reticulum. They're going to have, the biggest of which is going to be the nucleus. Their ribosomes are ADS ribosomes, and they have multiple linear chromosomes. These include your protists, things that we talked about, protists. This includes plants, animals, and fungi, okay, as well. Now, I want to talk a little bit about parasites. Parasites, these are, many of the parasites are going to be eukaryotes. Not every parasitic species. There are, like, for example, gonorrhea is this parasitic, so to speak, bacteria and viruses are obligate parasites, but many parasites that we think of when we say parasites, these are eukaryotes when we think about worms and things like that. Medical parasitology really does worry a lot about eukaryotes. Our protists, what we call the protozoans, fungal parasitic fungi. One of the most diabolical ones is the chytrid that parasitizes insects. If you guys are familiar with The Last of Us, the TV show based on the video game The Last of Us, is a cordyceps fungus. Arthropods are parasites, biting lice, bed bugs, ticks, things like that, biting flies. They also serve as vectors for lots of nasty diseases like chikungunya, Zika, and things like that. Platyhelminthes, these are flatworms. Nematoda, those are roundworms, things like that. So they can be, parasites can be multicellular, unicellular, and they can affect humans and affect many animals. Many parasites have a host they go into. That host then gets them into another host. Sometimes they get into what's called a blind host, where they cannot transmit to the next individual. So that happens. Protozoans are a really interesting group. They can be quite small, 5 micrometers to about 100 micrometers in diameter. All protozoans are motile except for the group Apicomplexa. They use pseudopodia, cilia, or flagella. That will be a thing I like to ask in my lab final. Is what do protozoans use for motility? Is pseudopodia, flagella, and a cilia. When I said it, I didn't say it in the order that I had it in notes, but there are about 50,000 known species of protozoans. Not that many cause disease, but they're definitely disease causing. They're usually tropical to subtropical, so usually in underdeveloped areas of the globe. But they are making their way to Florida, especially as Florida becomes more warm due to climate change. Now, they really affect the economic stability of these areas. People stay ill with parasites. They basically are weakened and can't work. So there can be significant economic instability caused by these things. A really good example of some of these protozoans that are parasitic are Giardia lamblia, that causes beaver fever, Toxoplasma gondii, Trichomonas pneumocystis, and Cryptosporidium. So there's lots of really well-known ones. I'll talk about a protozoan that's that affects like malaria and things like that. Protozoans that can be very difficult to culture because any host, any place that grows them usually just very hard. The species would fight them off pretty hard, but they're bigger, so your protozoans, so they are easy to see. So microscopy tends to be the best way of diagnosis. Serological tests are pretty difficult for some of them because we make a lot of antibodies against them. Typically, they have very complex life cycles. There's two distinctive stages. There's a cyst stage, which is dormant and resistant, and then there's the trophozoite, which is feeding and growing. A lot of times we can use the trophozoite in fecal matter as a way of identifying them, seeing trophozoites of like Giardia in fecal material. We can see that. I had a dog that I adopted from the animal shelter, and she had trophozoites. When I took her to the vet, she had trophozoites in her fecal matter. Protozoans, they go through different hosts, so there's many intermediate hosts before you get to us. Sometimes they infect what's called a blind host, and that host can't actually transmit. So we go to a primary host they want to get to, and then that poops out and it gets back to the next host. Many of these go through multiple hosts till they get to a primary host, and then they get sometimes they get into a blind host and we can't transmit it. One really nasty group is the helminths. These are multicellular. They belong to the animal kingdom. They are going to be in what we refer to, guys, as either the platyhelminthes or the nematodes. These are the worms that are parasitic that we really worry about. The type of helminth, they may be hermaphrodites, having male and female reproductive systems at some point in their life cycle. They may have a separate male, separate female, and they may even show what we call sexual dimorphism. So you may actually be able to tell this is a male schistosome, this is a female schistosome worm. So adult worms can actually be seen with the unaided eye. You poop them out and you'll see worms. Some of these worms about the size of a grain of rice, to some are larger. So because they have microscopic eggs and microscopic larvae, they are considered microbial parasites, though you can see the worms quite easy, the adult worms. Helminths tend to be really concentrated in underdeveloped areas. It was a really big thing to get them in Appalachia. People getting like especially pinworms, hookworms, and things like that. So bacteria is going to be our major thing. They are small. They're about 1 micrometer, 1 micrometer. That's a thousandth of a millimeter. It must be seen 1,000 times so we can actually see what shape it is and what arrangement. So they are very small. To see them, we're going to use compound light microscopes. Compound light microscopes can see things between 400 to about 1,000 times, where we have a source at the base that actually gives us the light through curved lenses. The lenses may be curved like this or maybe curved inwards that help us actually create a larger image. What we see is magnification. Microscopes that we use have 2 sets of lenses, so they're called compound. Okay. Now, when we look through it, we use what's called an ocular lens. The ocular lens is the lens you look through. They have a 10 times magnification. They're a set of 4 other lenses with a 4x, 10x, 40x, and 100x. The 100x is so powerful, light doesn't pass through it effectively. So we use immersion oil, or now we use, so immersion oil, we used to use a cedar oil. Now what we use is a water droplet, and that basically becomes part of the optic and enhances the vision, enhances the power. To find total magnification, something I will expect you to be able to do on quizzes and tests for lecture and lab, it is the multiplying the ocular lens by the objective lens you're using. For example, if I'm using a 40x objective, I have a 10x ocular lens, a 40x objective lens. 40 times 10 is 400, so the object I'm looking at is 400 times magnified. Okay, this will be something I expect you to do on quizzes, the lab final, lecture exams, things like that. Okay, I'll expect you to be able to do. Let's talk about bacterial cell morphology. Cells have morphology. This is the shape of individual cells. Now, because bacteria have rigid cell walls, they keep a shape. Their shape is pretty consistent and uniform, so generally the cells will be about the same size and they'll have the same shape when we look at them under a microscope. We have spherical rod and corkscrews. Spherical is referred to as a cocci. Singular, we call it a coccus bacteria. If it is a rod shape, we call it bacilli. Singular, we call it a bacillus. If these are curved, we call this vibrio. If they are spiral corkscrew, we call this a spirilla, singular spirillum. If they are flexible helices, we call that a spirochete. Then what they do is they have arrangements. And that's how they grow. Do they grow in chains? Do they grow in clusters? Do they grow in fours? Do they grow in eights, in 8 cube? How do they grow? Okay. We call this arrangement. So there's cell morphology, and then there is arrangement. Now, bacteria grow in a process called binary fission. The way this binary fission happens will result in these cells kind of being somewhat stuck together sometimes. If they are stuck together in a way, we call this an arrangement. Kind of like flower arrangements. The flowers are put together in a pattern. The cells are put together in a pattern. Now, one of them we get is called diplococci or diplobacilli. Diplo means they're in pairs. If it's strepto, it's in chains. Streptococci, streptobacilli. If it is in clusters, like grapes, we call this staphylo, like staphylococcus. Okay? If it is in 4, we call this a tetrad. If it's in 8, we call this sarcinae. I've got a little bit of a typo here. I did not get this I down when I was making my notes. This I should say, if bacteria don't have an arrangement, we call these singles. I'm always going to find little typos over the every time I teach it. I find something, I edit, I change, I adjust. I'm always working on my notes. So now this can give us, the arrangement can give us something about the species. We can help identify the species that we have. It can especially give us the name, like if it's a staphylococcus, a streptococcus, a bacillus, vibrio, cholera, things like that. So we have these genus names for species, sometimes have that. So we can have singles like Escherichia coli, streptobacilli, chains, tetrads, Micrococcus luteus, M. luteus, Staphylococcus aureus, growing in clusters. Okay. Now, one of the things we need to do is bacteria generally are actually quite clear. We really can't see them under a microscope. They don't have their own natural pigmentation unless they are photosynthetic. So most bacteria color us them pretty clear, so it's very hard to see them under a microscope. So what we do is we created many ways of coloring them called staining to make it see under a microscope, make them be able to be seen under a microscope. So we can get them colored just to see them. We can also color them in such a way that can help me characterize them down to help me identify species. One way to stain them just to see them is called a simple stain. A simple stain is just a stain to see, just for contrast. We call that a simple stain. Now, one of the things we do to stain them is we get our bacteria. Let's say I've got a petri dish and I got some bacteria growing on this petri dish. I get one of my inoculation loops and I pick up a colony and I put a drop of water on the side and I put my colony in and I smear that around and I make what is called a smear, a mix of water and bacteria, and smear them on my glass slide. Now, what we do is we smear that and we let it air dry. Then we generally heat fix it to the slide. And the heat fix is only done once the side is completely dry, then we heat fix it. We want to get, now, we don't want to get, it does kill it. Once it gets 50 to 55 degrees Celsius, it will kill it, but it makes them stick. And then when we rinse the stains off, the bacteria don't wash off. So then we can stain it. We can do a simple stain or things like that. Now, these are all basics of microbiology. What we're going to talk about coming up is what would have been the first lab if you were in person. I teach my online labs kind of a mix of we will do, this is what lab in person would do. Here is a close to what we're going to do lab simulation on Labster. And then here's a post-lab quiz. The post-lab quiz will be on my stuff. This is how I do it. You will get grades for doing your Labster work. You will get grades for taking my quizzes. And then there's lab exams. So the following is for procedure 1. Now, one of the things is aseptic technique is a very important thing we do in microbiology. In microbiology, one of the things I want to do is I want to be able to take and sterilely transfer. Let's say I've got a culture of bacteria that I'm given, and I've got some E. coli in a vial growing on in a broth, okay? And I want to make sure that I get that E. coli onto my Petri dish so that only E. coli is done. There's a variety of ways to do this. I'd have my Bunsen burner lit to have a cone of sterilization around where I'm working. I would sterilize my tools. I would wash myself. I would have washed the table that I'm working at. I would use a proper aseptic technique. I will clean the surfaces. I will clean my skin. I will use the Bunsen burner to keep the air sterilized. We will use what is called a trypticase soy agar, which is a media that gives everything a growing bacteria needs. It has vitamins, it has amino acids, growth factors, and everything that lets many bacteria grow. It is everything a lot of growing bacteria will need to survive. We put that in a Petri dish, or we put that in a tube, or whatever you want to grow it on. We can grow it in a media. It is food for bacteria. Now, what we basically would have you guys do in lab is we would get what's called TSA plates. We get 2 of them. One of these TSA plates are called the air plate. And what you do is you take the lid off and you let it sit in the lab for a certain amount of time, till pretty much you're ready to leave. Then you would close it, and we would label it and things like that. And on one plate, we just leave it open until it's time to go. What's happening is bacteria and microbes are floating in the air, and they're going to fall and settle on your plate. And then you're going to put the lid on it, put it in the incubator, and see what nasty things would have grown. The other one we divide into 4 quadrants. Okay, we 1, 2, 3, 4. Now, when we do Petri dishes, always label not on the lid, but label things on the side where the agar is. And what we do is we label it. We make our 4 corners here, our 4 little quadrants. One of the things you would do is you would take your thumb and push it onto the agar. Then you would wash your hands, following the CDC hand washing methods. Then we would put our clean thumb on the agar. We start with dirty hands, and then we make sure we don't tear the agar. Now, we do the second one, but we wash the dirty thumb. We wash our hands. Now we do the same thing. For number 3, we get what's called a fomite. What is a fomite? A fomite is something I can pick up and take with me relatively easy. This is a very common fomite. Fomites are things that I can pick up and take with me easily that might carry pathogens on them. For example, my phone. Okay? Step 7, we swab some surface, like a keyboard or toilet seat, something like that, a chair or whatever. I've had students run over to the food service area and swab the tables that people eat at over here at Metz. Back for that. Actually, it was Subway when I last taught the lab in person. It's been a little bit. We hired Dr. Sarah Hill, and I wasn't needed to teach micro as much because she primarily taught micro. But when I was teaching micro, and then I'm back at it again, which I really enjoy. I'm really passionate about it. I really enjoy it. So anyway, that's what we would do there. Then there's a big thing. We would normally get tested on this, but we have to do it differently in an online lab. So in an online lab, I'm doing a midterm and a final, and weekly quizzes, and your labs are labs. Now, what we would get is to sterilely move from one broth to another. Normally, you would have to do this in front of me for an exam. You would take to sterilely transfer broth from tube A to another tube B. You would need goggles and gloves. You would need to be using a Bunsen burner and an inoculation tube. You would learn to use the pinky finger technique, where you hold your pinky finger and loosen the caps and things like that with your pinky to take your caps on and off. Okay. You would use your dominant hand and hold your pinky and do like that, and hold it with your pinky finger and turn it. We call that the pinky finger technique. You would so we would open it up, and when we did that, we would flame the tube. We would get a loop full of broth. We would flame the tube and cover it. We would then open the next, flame the tube, inoculate, get that out, flame the tube, and close, and put it up. And then we would put that into our incubator and see if you got any bacterial growth, if you properly did this. And that would be part of the grade, is that you got no growth. You do the steps right, and you don't contaminate. And that's appropriate technique. Then we would also do this by going from a broth to a plate. Same thing. Every time you open it, you flame the tube, you flame your loop and all that, and run through it. And what we do is we would put broth from the tube onto the plate. Now, one of the things we do is we want to make sure that we are minimizing our plate. So one of the things we do, and if I just had thought about it, give me 1 second. All right, with the magic of the pause button, I showed up and I grabbed, in my desk, I have a sleeve of non-sterile Petri dishes. So one of the things we would try to do is keep this where things won't get in, and inoculate in such a way that we minimize any kind of things. One of the things people will do is they will dig in and tear the agar sometimes. So this is your typical Petri plate. You put your media in it. There's a smaller lid and a bigger bottom. And what we do is we always label on the bottom where the media is, because this lid can turn. So, and I've got a video that you can see how to do all this. I've got videos posted. All right. Well, this is the end. So I hope that helped you guys. There's a quiz on this, so please take it. And we'll see you guys in the next one.